Marshall Nirenberg was born April 10, 1927. He went to school at the University of Florida and he received his Bachelor's of Science degree and in 1952 a master's degree in zoology. He then became a research biochemist at the National Institute of Health. He continued on to receive many prestigious awards and honors. Philip Lider was born November 19, 1934. He went to school at Harvard University and graduated from Harvard Medical School and completed his residency at the University of Minnesota. These scientists discovered that 3 nucleotides form a codon. A 3-nucleotide codon produces 64 combinations. In 1961, Heinrich Mette and Nirenberg started testing the triple codon hypothesis. They used a cell-free E. coli because they thought the extract could contain all the components needed to translate mRNA into proteins. This extract was treated with DNase to destroy any remaining E. coli DNA. They then made synthetic mRNA composed of entirely uracil and added this to the extract along with radio-labeled amino acids. When they examined it, they found polypeptides entirely composed of phenylalanine, which caused them to find out one of the 64 codes, UUU. They then conducted other research and created a template with other nucleotide combinations. Lider then came in and finished the experiment, because the codes were sometimes hard to decipher. He used the activation of tRNA to solve the problem. The key step of the experiment was that they labeled one type of amino acid at a time and put the mixture through a millipore filter. The sample was then tested for radioactivity. If there was radioactivity found in the sample that did not pass through the filter, the corresponding amino acid had been added. This discovery occurred in 1964. Before this experiment started, George Gamow suggested the genetic code was made of 3 nucleotides per amino acid. However, he proposed that the triplets were overlapping. Seymour Benzer in the late 1950s developed an assay using phage mutations which provided the first detailed linearly structured map of a genetic region. The next discovery made from this scientist's discovery was Corona's experiment. He verified the entire code.
And I thought that maybe a triplet, 3 bases alone, might be recognized on ribosomes by the appropriate species of transfer RNA with the appropriate amino acid attached to it. And I tried this, and the very first experiment worked. And so we used a very simple, different kind of assay, a very simple assay to determine the base sequences of codons by measuring the binding of radioactive amino acid attached to transfer RNA, which bound to ribosomes and recognized a triplet, 3 bases. And we could separate the complex from the unbound amino acyl tRNA by adsorption on filters. Very simple assay. It worked.